So thank you very much for coming to today's session of the Ensembl webinar. So I'm Victoria Newman, and I'm a member of the Ensembl outreach team. And today I'll be presenting the variation data that we show in Ensembl. So the objectives of today's session are to provide more context for the Ensembl genome browser and what it is, to show you the types of data that you can find in Ensembl and how to find them, so how to navigate the Ensembl browser website, and also, importantly, where to go for help and documentation as you start using Ensembl in your own research and want to build beyond what we cover in these sessions. So we are today in the fourth session, which will cover variation data in Ensembl and the Ensembl VEP tool, which stands for Variant Effect Predictor. You may already have seen the first three sessions. So in the first one, my colleague Helen presented an introduction to Ensembl. After that, my colleague Emily explained how we annotate our genes and transcripts. And then last week, I presented the Biomart data export tool. Next week, we'll move on to my colleague Ben, who will present comparative genomics analyses. So today, I'll begin with a presentation on variation data. And then I'll have a live demo on the website to show you how variation data appear in the browser. And then I'll go back to my presentation and I'll introduce the VEP tool to you. And then we'll have another demo which will focus around using the VEP in the browser. Lastly, there are some exercises available to you, and I'll show you in a second where you can find them. So in case you have any questions, we have muted all the mics, but please don't hesitate to ask them. So you can use the chat box here in the webinar interface, and my colleagues will reply. So I have my colleagues Ben and Emily here with me. Because there's no threading, if you do need to follow up to the response that they give to you, please do add at Emily or at Ben in your reply so that you can just continue your discussion. So we have a dedicated page available in the training site of the EBI. So the EBI is Ensembl's parent institution. So if you go to this link, you'll see all the materials from the webinar series that we're doing. And you can access the different sessions in the menu on the left-hand side of these pages. So each session it will have its own page, and once we've actually uploaded the video of this webinar, it'll be available on this page. It'll be hosted in YouTube, and we also have a link to Youku for users who can't access YouTube. And in addition, we'll have a PDF of the slides available, so you can download them and look through them at your leisure. We also will make exercises available, and you'll be able to get to the exercises either through the left-hand menu or just by clicking the links to navigate through the pages. And there will also be answers. So in case you want help with the exercises, again, there are answers on the page I just showed you, but you can also go ahead and join our Facebook group. So the Facebook group is available to participants in this webinar, and you can have a joint discussion about how to arrive at solutions. There's information about how to join the Facebook group, again, on the page for the online course. In case you have more nuanced questions or you're not on Facebook, then please do go ahead and email us at helpdesk@ensemble.org with any questions that come up, and we can try to help you out. Okay. So this takes me to today's presentation. So again, we'll be talking about variation data in this part of today's webinar. So I'll start just by giving you a brief overview of how we classify variants in Ensembl, and I'll tell you a bit about the species for which we have variation data and also where those data come from. And then we'll go to the browser and I'll show you variation data in the Gene tab, the Location tab, the Variant tab, and the Transcript tab. Then we'll have another brief presentation and a demonstration on the website to show you how to use the VEP tool, so the Variant Effect Predictor. So broadly speaking, we classify variation into two types: small scale and large scale. So small scale variation can affect one or a few nucleotides, but according to the definition that we use in Ensembl, it will affect 50 or fewer base pairs of sequence. Large scale variation can affect an entire genomic region, but we classify it as variants affecting more than 50 base pairs of sequence. So small-scale variation can include things like small insertions and deletions, or single or multi-nucleotide polymorphisms. So the terminology, you might have seen some of these terms before, so things like dips and indels, or SNPs, or MNPs. So if you consider any two individuals in a population, they might differ in a few loci. So if you compare the sequences of these two individuals, you can see that there's a SNP here where they differ in a single nucleotide. As you read through the sequence, you can see that there's an indel. So one individual has a base inserted with respect to the other, and then later on deleted with respect to the other. And we also have another SNP. So large-scale variation, again, is affecting larger genomic regions. It can also be referred to as structural variation. And so examples of large-scale variation are things like copy number variation, or larger deletions, duplications, or amplifications of sequence. And large-scale variation can also involve chromosomal insertions or translocations. So I've got an example of copy number variation on the left here. So if we consider that the reference sequence has 4 genes in a row, these differently colored rectangles, here's an individual who has a duplication in the red gene, and here's an individual who has a deletion in the green gene. So if we think about the chromosomal scale, let's say we have 2 non-homologous chromosomes here. We have A and B, and this is how they normally look. So here's an example where A has a duplication or an amplification of this orange region. And here's an example where A has a deletion of the orange region and possibly some additional sequence. Insertions can happen when sequences from a non-homologous chromosome recombine into another chromosome. So here we can see that we have the introduction of sequence that came from B, so this dark gray sequence, into A. And translocations can occur when non-homologous chromosomes swap ends, for example. So here we can see that sequences from chromosome B have landed on chromosome A and vice versa. So in principle, variation can occur in any genomic position, but in Ensembl, we like to think about the consequences of variance in relation to genes. So for example, you can have variants that affect coding sequences, and they can be either synonymous or non-synonymous. So synonymous variation means that there's no change in the protein sequence encoded by the gene. Non-synonymous variation results in a change in the protein sequence encoded by the gene. So it's also referred to as missense variation. You can also have variation that lies elsewhere. So you can have variation that's upstream or downstream of a gene. And one example of this type of variation might be variation overlapping a regulatory region. So it might modulate gene expression. You can also have variation that lies outside coding sequences, but within the sequence of the gene. So it can fall within the 5' or 3' UTRs, the untranslated regions, or in an intron. And you can also have variation that affects or disrupts splice sites. So because there are a broad variety of consequences that can result from variation, we use a controlled vocabulary and ontology to describe these possible consequences. So in one of the previous sessions of this webinar, we talked about the gene ontology. So here we're using the sequence ontology, which is just another ontology to describe the consequences of variation. So like a normal ontology, there are accession numbers that refer uniquely to ontology terms and their definitions. And so we just mentioned missense variation. So you can see here the sequence ontology term is missense variant, and the definition is provided. So it's given as a sequence variant that changes one or more bases, resulting in a different amino acid sequence, but where the length is preserved. So again, the protein sequence changes, but the length of the protein doesn't change. And you can also see synonymous variant here. So again, a sequence variant where there's no resulting change to the encoded amino acid. So because missense variants lead to a change in protein sequence, they might also disrupt protein function, or they might alter protein function in some way. So in Ensembl, we use a couple of algorithms called SIFT and polyphen to score the changes in amino acid sequence to tell us, in essence, whether protein function might be preserved or might be disrupted. So SIFT in particular is looking to see how well conserved an amino acid is in relation to homologs of the protein in other species. And it's also looking to see how well conserved the domain in which the amino acid that's altered is. So if, for example, we just think about homologs of proteins, if we have a region that's highly conserved, so these green blocks represent highly conserved sequence, it could be that variation that falls within those regions, those highly conserved regions, will be less tolerated because they have an important function to play. Polyphen considers conservation, and it also looks at the actual chemical change in the amino acid. So amino acids all have side chains, and these side chains can vary from amino acid to amino acid. So for example, some of them are charged and some are neutral, and some are bulky and some are very small. So depending on the change to the amino acid that results from a missense variant, you may disrupt folding of the protein. You may also disrupt protein-protein interactions with binding partners, or you may perturb a residue that's functional, for example, that's modified in some way, or that plays a catalytic role. So polyphen considers changes to amino acids in addition to conservation to guess whether or not a missense variant is likely to be tolerated. And you can read more about SIFT and polyphen in these two papers. So SIFT and polyphen are predictive. They're not based directly on experimental evidence. So we do suggest that you sort of interpret their predictions cautiously. They both use scales from 0 to 1 to indicate whether variants are likely to be tolerated in a cell or tissue or deleterious. But unfortunately, the scales are opposing. So we color code them, and we would encourage you just to look at the colors. So red is bad and green is likely benign or tolerated. So in Ensembl, in the main Ensembl site, we have variation data available for 22 species. And we also have variation data available for a variety of species in Ensembl genomes. So these are species in Ensembl plants, fungi, protists, and metazoa. We don't have variation data available for Ensembl bacteria because our database is simply too large. You can find more information about our variation data at this link. And we import our variants from a fairly wide variety of sources. Again, you can find more information about the full list of repositories from which we import variants at the link. And you can also find information at this link about the repositories from which we import phenotype information. We principally import short variants, so sequence variants from the dbSNP repository, which is enormous. And we also have structural variation data, for example, for some organisms that we import from Decipher. So Decipher focuses on human and DGVA, but this is just a minor subset of the repositories that we draw from. So in addition to variants, we also report the frequencies of those variants. So, for example, the HapMap project looked at a genetic variation in the human population, and HapMap used genotyping arrays to assess frequency in individuals from 11 populations worldwide. So the idea was just to explore genetic diversity in the human population. So because HapMap used genotyping arrays, it recovered the frequencies of known variants. We also report data from the 1000 Genomes project. So the 1000 Genomes project built off HapMap, and it used next generation sequencing to assess variant frequency in individuals from 26 populations globally. And because it used next generation sequencing, it was able to capture the frequency of known variants, but also to discover novel variants. Okay, so how do variants actually look in Ensembl? So I think my colleague Helen showed you just this, this diagram. So in essence, what we're showing here is that in the reference sequence, the individual chunks of sequence, which are called contigs, come from individuals. So individuals are fairly outbred. So when we talk about the reference sequence, we're not talking about the consensus sequence. If we're talking about the human reference genome, we're talking about a genome assembly that contains sequence from a number of different individuals, but individuals who are genetically idiosyncratic. So if we look at a run of sequence in one contig, so let's say this particular contig, let's say we have a T at this position. So let's say that actually in the population as a whole, T is relatively uncommon. And let's say that actually all primates have a G, which is also the more common allele in the human population. And let's say that, in fact, T is a disease-causing allele. Even so, because T is the allele in the contig in the reference genome, it's referred to as the reference allele, and G is the alternate allele. So the alleles will be written T/G. They'll be described as reference/alternate. So the reference allele doesn't have it doesn't have any bearing on whether it's normal in the population, if normal is really an appropriate term to use in this context. So what happens when we're talking about a gene that's transcribed from the reverse strand? So if we look at the genome here, on the forward strand, we have reference/alternate, so we have T/G. And on the reverse strand, we have the reverse complement of T as the reference. So we have an A as the reference allele, and we have a C as the alternate allele because it's the reverse complement of G. So if we're talking about a gene that's transcribed from the reverse strand, then in the actual sequence, the sequence of the transcript, the reference will be an A, and the alternate allele will be a C. So in the Ensembl database, we will always describe alleles with regard to what's on the forward strand. But if you're looking directly at the sequence of a gene or transcript, you will see the sequence in the reverse strand. So I'll show you that in a second when we go across to the browser. So just keep in mind that in Ensembl, we curate alleles to make sure that in the database, we're always describing what's on the forward strand. But if you look in the repositories from which we draw these alleles, from which we draw these variants, you may see that they're reported in a sort of a variety of ways. And in the literature, you'll also see that alleles are reported in a variety of ways. And typically in repositories or in the literature, variant alleles will be described just as they're reported by researchers or authors. So they may not agree with what we say in Ensembl, but they mean the same thing because we have undertaken this curation process to standardize how we report them. Okay, so we're going to go across to the browser now, and we'll look at the MCM6 gene. So we'll look at variants in the gene, and we'll also look at variation data in the genomic regions surrounding the gene. And then we'll explore one particular variant to find more information about it. So I'll just close my presentation. Okay, and then I am going to just go across to the browser now. So you've already seen this page a couple of times, so I'm just going to go ahead and search MCM6. Okay. So the human MCM6 gene comes up first in the search results, so I'll just click on this. And so again, this takes me to the gene summary in the gene tab. So if you look in the left-hand menu, you can find the information that is reported on this gene. So the first thing that we'll do is we'll go to the MCM6 sequence. And so we can look at variant markup in the sequence. So this is just the sequence of the MCM6 gene, as you can see. And I want to show you the FASTA header just to start out with. So we're reporting that we're looking at the GRCH38 version of the Human Genome Assembly, and we're on chromosome 2. And the gene lies between these two coordinates, but it's on the reverse strand. So there's a -1, so the MCM6 gene is transcribed from the reverse strand. So what I'm going to do is configure the page just by clicking on this button. And so I am going to show variants. So if I turn this on, we'll just see all the variants that have been reported in the repositories from which Ensembl draws variation data. So you can get a sense of where they lie within the sequence of the gene. Okay, so sometimes this takes a while to load. So just notice that the variants are color-coded. So these, again, are using sequence ontology terms, and we apply sort of standardized color coding to describe the variants just as a shorthand, so you can get a sense of what they are without too much effort. Okay. So as we scroll down here, we can see that we have some variants lying within exonic sequence, and we also have variants lying within intronic sequence. So again, these variants, so the gene is transcribed from the reverse strand. The variants are being described using IUPAC nomenclature, so you can see that there are some symbols that clearly are not nucleotides. So if I just mouse over this, now I'm seeing that the reference allele in this position is a G and the alternate allele is a C. So we don't show you the actual alleles when we turn on variant markup, but if you just mouse over any variant, then you can see what the alleles are. And so these alleles are described with regard to the actual sequence of the gene as it's transcribed from the reverse strand. So G/C in the case we just looked at. Okay, so you can turn on variant markup. You can also explore variants just in tabular form by going to the variant table, which is accessible at this link. So this shows you a table of every variant that has been observed in association with this gene. And typically, there's sort of quite a large number of variants in the variant table. So we have filters available so that you can restrict the table to variants that are interesting to you. So you can filter by consequence. So for example, here, you can filter to see only missense variants. So if I turn all off, then I can come here and turn on Missense variants. So that's 918 variants that are known to affect the sequence of the protein encoded by MCM6. And okay, so my table has now filtered, and you can see that these missense variants all have SIFT and polyphen scores associated with them. And you can also see that SIFT and polyphen disagree in a couple of instances. So again, they are predictive, and so you should interpret them with caution, usually. And you can filter in other ways as well. So for example, if you want to restrict the variants that you're seeing by genomic location, or by the coordinates, the amino acid coordinates within the protein, you can apply additional filters. Okay, so that's the variant table. And you can also look at the phenotypes that have been associated with this gene just by clicking here at left. So I'm not going to show you that now. I'm going to go to the genomic location surrounding MCM6 just by clicking up here to go to the location tab. So we've already seen the location tab, and you know that you can configure the page, again, by clicking the Configure this page button. What we'll do is we'll turn on a few tracks that contain variation data. So I will just, so actually you can see these are active tracks, and you can see that we already have the track showing all short variants detected in the 1000 Genomes Project turned on. We also have all phenotype associated short variants, and we have structural variation data from the 1000 Genomes Project. So let's just turn on a couple of other tracks. So I am going to turn on common short variants from the 1000 Genomes Project by clicking here. And so I'll just turn it on in normal. And then I am also going to turn on larger and smaller structural variants from all sources. So, okay, so here we have structural variants. So I'll turn on smaller structural variants from all sources in expanded, and I'll turn on larger structural variants from all sources in compact, let's say. Okay, so these tracks will load in the window. So here we have all short variants from the 1000 Genomes Project, and we have common short variants from the 1000 Genomes Project. We also have phenotype-associated short variants, and then here we have the structural variants, so the smaller structural variants we just turned on, and here we have the larger structural variants we turned on. We also have the structural variants identified in the 1000 Genomes Project. So you can see we do have some structural variants that are covering quite a lot of sequence in both the smaller and the larger. If you want more information, we do have a variant legend, again, with color coding. So these, again, are sequence ontology terms that are describing the types of variation. And we also have a structural variant legend that shows you what the color coding stands for. So these are all clickable, and if you want more information about any one of the variants, you can just click so on the short variants or the structural variants, and clicking brings up a pop-up. It tells you the code that's been assigned to the particular variant, and it also tells you the genomic location. It tells you where we imported it from. It tells you the class of variant, so in this case it's a copy number variant, and it's a deletion. Okay, so at this point we are going to go across to the variant tab to look in more depth at one variant associated with MCM6. So I'm just going to search for it because it would be hard to track down in the midst of all the variation we've seen associated with this gene. So my variant is RS 4988235. I think 4988235. Yep. Okay. So we'll search for that, and in the search results, again, we have just one hit. So clicking on this link takes us to the variant tab, which essentially just collates all the information we have on this variant. Okay, So the first thing that you can see is that its consequence is an intronic variant, so it doesn't lie directly within the coding sequence. And you can see that the reference allele is a G and the alternate allele is an A. So I'm not sure where I'm going with this. So in any case, in the reference sequence, the allele is a G. And you can see that A has an allele frequency of roughly 16% in the population. Okay, so you can see that it's on chromosome 2. And here the coordinate is reported with regard to the forward strand. And also the reference and alternate sequence here is reported with regard to the forward strand. So there are a number of links that are available on the left-hand side, and you can explore the same information by clicking on these buttons here. So first we'll go to genes and regulation. So genes and regulation will show us the genes and transcripts that this variant is associated with. So you can see that it is associated with 3 transcripts, and they're all in the MCM6 gene. These transcripts all belong to the MCM6 gene. So what you can see in this table, so you can see the identity of the transcript, and then you can see the strand. And so the strand, again, is shown by a minus. So we saw previously in the gene tab that MCM6 is transcribed from the reverse strand. So the allele is an A, but the transcript allele is a T. So the allele is reported with regard to the forward strand, but because the transcript, because the gene is transcribed from the reverse strand, the transcript is given as the reverse complement. The transcript allele is given as the reverse complement of A or T. And again, you can see the consequences. In each case, the consequences of the variant alleles do not implicate coding sequence. Okay, so that was genes and regulation. So we can also see what populations this allele has been reported in. So if we just click on population genetics at left, we can see information at top from the 1,000 Genomes Project. So we have these little pie charts up here, and so these show us, first of all, the frequency of the allele in the entire 1,000 Genomes cohort, so that's about 2,500 individuals. And then we have these superpopulations that cover different continents, so Africa, the Americas, East Asia, Europe, and South Asia. And so what we can see is that the allele occurs at different frequencies. The variant allele, A, occurs at different frequencies in the different superpopulations, but it's most common in the European superpopulation. And you can see a breakdown in this table here of the actual frequencies at which the allele was observed in the subpopulations within each superpopulation. And you can also see the genotypes that were observed. So if we look in the African populations, we do pick up some individuals who are homozygous for the variant allele in certain subpopulations. And we also see some individuals who are heterozygous for the variant, but in principle, most people are homozygous for the reference allele. And then we can see the frequency changing. We see quite a number of people who are homozygous for the variant allele in different European subpopulations. Okay. So now the question arises as to what phenotype is linked to this variant. So we can click on phenotype data to find out. And so this just loads a table that reports different phenotypes that have been associated with the variant. And so we can see that one of them is lactase persistence. These are people who have the variant allele are able to tolerate lactose as adults. We can also see the phylogenetic context. And so this just tells us whether the variant allele has been observed in primates other than human. So here we're just looking at 8 primates, which are all listed here. And so what you can see is that the reference allele in human, again, is a G. And in the primates that we're looking at here, they also have a G at this position. So the variant allele A leads to lactase persistence in adults. So what we know is that there's some proportion of human adults who can tolerate lactose as adults. And primates don't seem to have an allele that confers this phenotype. So what we can say is that the ability to tolerate dairy products in adults seems to have arisen in the human population. So that's just an example of the types of variation data that we have in Ensembl and the sorts of information that you can find in the Variant tab. So I'm going to quickly go to the Gene tab again for MCM6. So I'll go back to Genes and Regulation. So in fact, I'll go directly to a transcript page because I want to show you the transcript haplotype function that we have. So in essence, variants do not tend to in Ensembl, we describe single variants in many cases, but in the population as a whole, variants may be linked in the germline and they may be co-inherited. And so by looking at haplotype data, we can see which variants tend to be co-inherited. So the Ensembl variation team have made predictions about co-inherited variants by looking at 1,000 genomes data. And so here I'm showing you a table of protein haplotypes, so linked variation. And you can also switch this table to look at the nucleotide sequences for these variants. So what you can see is that we have a series of variants that occur that affect single amino acids. And here we also have an example of two amino acids that tend to be altered together. And you can also see the frequencies of these variants, and you can see the frequencies in the 1,000 genome superpopulations as well. So this table just gives you an idea of whether or not variants have a tendency to be co-inherited, and in addition, what populations globally these linked variants are found in the cases where they are linked. Okay. So that ends the first demonstration. So I'm just going to go back to my presentation. And now we will talk about the VEP. Okay, so the VEP stands for Variant Effect Predictor. And this is a tool that was developed by my colleagues here at Ensembl that lets you predict the effects of variants that you yourselves discover in the course of your own research. So what you can do is you can input variants into this tool, and they can be any type of variants. So they can be sequence variants or structural variants, SNPs, insertions, deletions, copy number variants, or again, variants that affect a larger genomic region. And you can input them using a variety of different notations. So you can just input the genomic coordinates. You can upload a variant call format file. You can input HGVS notation, or if you want to explore known variants, then you can just you can input their repository IDs. So, for example, a DB-SNP ID. You input variants into the VEP, and you spit out all kinds of different information. So first, you can see the gene, the transcript, or the protein, which the variants are associated with, if any. If they're missense variants, you can get information about possible functional implications, so SIFT and polyphen scores. If the variants are already known, you can see at what frequency they've been observed in different populations, and you can see if they overlap regulatory regions or splice sites. And then lastly, If they've already been reported, or if they coincide with variants that have already been reported, you can pull out literature citations describing these known variants in PubMed. So we already have a number of species in Ensembl that we have variation databases for use with the VEP. This is just a graphical representation of the species in the main Ensembl site, and we also have VEP databases for a variety of species in Ensembl genomes. So what happens if you want to use the VEP to predict variants in a species for which we don't already have a VEP database, like T-Rex? So what you can do is you can set up a cache. We have different versions of the VEP, including just a script that you can run from the command line, and you can build an offline cache which will speed up the running of this script. So we have pre-built caches for the species that I showed you on the previous slide, but you can also make your own cache just from GTF and FASTA files. So in other words, the VEP is compatible with all species, even those for which we don't directly have variation databases available. And there's more information on creating a cache at the link. So today I'll be showing you how to use the web interface of the VEP, but we also have a standalone Perl script, again, that you can run from the command line. And this is appropriate if you're more of a power user. And the VEP has various plugins that are available for use with it. So the plugins just can add extra functionality, so they can draw on external data or external code, and they can extend, filter, or manipulate VEP output. Okay, so we'll go back to the browser now, and we'll run the VEP. And so what we'll do is we'll just explore 4 variants on human chromosome 9. So one of them is a deletion of a single nucleotide at this position on chromosome 9, and then the other 3 are SNPs. And we have the information about the genomic positions again. So we'll use the VEP to see whether these variants have already been annotated. We'll see which genes are affected by the variants, and we'll also see whether any of the variants lie in regulatory regions. So I'll just exit my presentation again, and I will go, actually, so I have pre-copied, what am I doing here? So I've pre-copied the notation into the footer here. So I'm just going to copy this so I can input it into the VEP. Okay, so where can you find the VEP? It's at the tools link here in the navigation bar, which I already have loaded. So I'll just click on this again. So it loads a page like this, and the VEP is the top link in this table. Okay, so this is the web interface of the VEP here, and the species has been preset at human, which is what we want. So you can paste your variant data into this box, and if you're not quite sure of the appropriate notation, we have examples that are down here, and by clicking on any of these links, you'll just paste an example of the notation into the box to serve as a prompt. So I'm just going to copy what I brought from my presentation, but this is only the first 3 variants. So I'm going to get the information for the 4th variant, and I'll explain to you what I'm doing as I type in the relevant information for that variant. Okay, so the 4th variant is a G to an A substitution, and it's at this locus. So okay, so our variants are on human chromosome 9. So the first coordinate is the chromosome. And then the second and third pieces of information we need to put in are the bounds of the variant. So if it affects a single base pair, then the start is the same as the end. So I'll just paste this coordinate twice. So we have chromosome, and then we have genomic start coordinate for the variant, and then genomic end coordinate for the variant. And then the variant is a G to an A substitution. So next I will enter reference /alternate. So it's a G to an A substitution. we're reporting it with regard to the forward strand, and we'll just call it var4 so that we can interpret the results in the results table that we get out. So what you can see here is that at the top, we have an A in the reference, which has no base in the alternate. So we're talking about a deletion of an A at this position on the forward strand. And then these all represent substitutions with, again, the reference allele being given first, and then the alternate allele being given second. Okay. You can also upload a file in case you have a longer list of variants that you'd rather not paste in. We're using Ensembl default notation, but again, there are examples of other Ensembl default notation and VCF format, as well as database identifiers and HGVS notation here when you click on the links. Okay, so first we can choose which transcript database we want to use to associate our variant with transcripts. So I'll just keep Ensembl. And then we can see, for example, identifiers and frequency data. So here we're reporting the gene symbol for any genes associated with the variant, but you can also opt to see CCDS IDs, Ensembl protein IDs, UniProt IDs, and so on and so forth. Okay, so frequency data. Find co-located known variants is quite useful. So what this does is it tells you whether variation in the same region has been identified. So in our particular cases, we're talking about SNPs. So the VEP can report whether the same variant or another substitution at the same position has been observed. That can be interesting just in the context of knowing whether or not this is sort of a site that sees a lot of variation in an organism. You can also report frequency data for these variants. So we can see frequency data from the 1000 Genomes Project, for example. And we can also retrieve the IDs of any citations in PubMed of co-located variants. And then in miscellaneous, we can report the transcript biotype. So for any transcripts that are associated, are they coding, are they non-coding, and so on and so forth. We could see protein domains. So let's say the variant alter sequence in an SH2 domain of a protein, we would be able to have the VEP report that information. You can find what intron number or what exon number the variant lies in, and then these are the flags that indicate our level of confidence in the particular transcript prediction. And then you can also enable pathogenicity predictions for Missense variants, so SIFT and polyphen scores, and we have plugins that allow you to report pathogenicity predictions for a few algorithms besides SIFT and polyphen. We can also see whether our variant overlaps predicted regulatory regions or splice sites. So I'm just leaving the default settings, and I'm going to go ahead and press run now. And so this will generate a table. It's queued at the moment, so typically it doesn't queue for very long. And this just refreshes automatically every 10 seconds. So hopefully we don't have too many jobs running at the moment, so we can retrieve our results relatively quickly, but we'll just see. Okay, so it's done already. So to see the results, you would just click here. And so this is going to generate a pretty large table. So first we have just a summary of our job. So we can see that all 4 of our variants were processed and none of them were filtered out. And 1 of our variants was novel, but the other 3 were pre-existing. So the variants overlap 2 genes and 7 transcripts, and in fact 1 of them does overlap an annotated regulatory feature. So we have a pie chart that breaks down all the consequences that have been observed. And for the consequences falling within coding regions, some are synonymous, but the majority are missense. Okay, so now we have this table at the bottom here, and unfortunately, it's going over to the right. So I'll just quickly show you. So I'm going to filter, I'm going to sort by the variant number. So now they're just listed in numerical order. So you can see the name that we've assigned to the variant. You can see the genomic location that we input, and you can see the allele. So the first one that we input was a deletion, and then the other ones were SNPs. So the VEP is reporting the allele, and then it reports the consequences, again, using these terms from sequence ontology. You can see the gene that's overlapped, and you can see the Ensembl gene ID. And you can also see the feature type. So most of the variants are affecting transcripts, but we see this one is a regulatory region variant, and it overlaps a regulatory feature. So you can also see the Ensembl feature ID. So there are ENSTs, or transcript IDs, for the majority of the features, but we have an ENSR, which is a regulatory region ID for the regulatory feature that's overlapped. And you can see that it's a promoter. And so again, we have the biotypes, so we can see the biotypes of the transcripts, and we can also see the biotype of the regulatory element. So we're seeing as well which exon these variants lie within, if any, or which introns they lie within. And we can see the positions in the cDNA, in the coding sequence, in the protein where applicable, and we can also see the amino acids in the protein where applicable. So again, we have reference and then alternate. And you can see the position in the codon that is actually altered by the base change in the genetic sequence. If the variant is pre-recorded, you can see the ID that's known. So these are DB SNP IDs. So you can see that this variant is the novel one because there's no known ID. However, in the rest of the table, you can see that there are known IDs. And then as we just scroll over further, you can see additional information. So this is a Missense variant, and you can see the SIFT and polyphen scores. And you can also see the frequencies for the variants that were picked up in the 1000 Genomes project. So this is a bit of an overview of how the VEP works and the types of data that you can get out of the VEP. So you can sort the columns in this table, you can hide columns that you're not interested in, and you can also filter the table in case you only want to see particular results. And you can also go ahead and download the contents. You can also directly export lists of variants or genes to Biomart for further study. So I'm just going to go back to wrap up my presentation quickly, since we're running out of time a little bit. So again, you can find help and documentation both on the training site for the EBI and also directly on the Ensembl website. And do check out our YouTube and Youku channels in case you want to find additional content, just with short videos that explain different functions and data within Ensembl. Don't hesitate to email us directly at the help desk if we can answer a question for you. And do subscribe to our dev or announce mailing lists if you want to keep up with what's going on. And again, you can submit more technical queries involving essentially involving programmatic queries to the dev mailing list, whereas announce is just used to track what we're up to. Do follow us on Facebook and Twitter, and do also check in on our blog to see what we're up to from time to time. And do cite our publications, please, if you use Ensembl in your research. So the most recent publication from the Ensembl project is this one, and we have a couple of publications that walk you through different aspects of the browser, which you can also refer to. So again, I'd just like to show you the entire Ensembl group and to thank everyone in this group for their hard work on the Ensembl Genome Browser. And I'd like to thank all our generous funders on whose support we rely. Thank you very much for your attention. Do check out the exercises and come back to us if you have any questions. And with that, I'll just close.